Good morning, everybody. We are going to study today the expression of genetic information. Yesterday we explained a little bit how one gene was able to produce a protein, and we talked about two processes. We talked about transcription and we talked about translation. Why? Because one gene cannot synthesize or produce directly a protein. It has to go through these two processes. Okay? Transcription is going to be inside the nucleus. Okay? Just as replication. Right? But translation, that we will study it in the next video, that is not going to happen inside the nucleus. That is going to happen in the cytoplasm. So when this transcription process finishes, the RNA messenger that is going to be produced is going to get out of the nucleus. Okay? And it's going to go to the cytoplasm. There, ribosomes will work and transcription RNA will work. Transfer RNA, I'm sorry. So how does this work? One important thing that it's not on the book is that enzyme responsible for the synthesis of the RNA is going to be RNA polymerase. We talked about polymerase before, but we talked about it in replication. Then it was DNA polymerase, okay? Because it was going to produce a strand of DNA. But here is going to be RNA, because in this process what we are going to get at the end is a strand of a messenger RNA. One thing that you should know about transcription is that it's not just messenger RNA that is produced. For example, the transfer RNA is also produced here, and all the RNAs that are in your body, they are going to be produced with the same manner. ¿Vale? It's exactly the same. But we are going to focus on messenger RNA. Okay? Because it's important for the following process. Okay. So what do we have? We have our double helix strand of DNA. What has to happen? It has to open, because if not, it's not possible to produce anything inside here. So again, there are some enzymes that are going to open these two strands and separate them. Okay, so you have these two strands, and here you have the nucleotides. Remember that I only put the nitrogenous bases because the rest of the nucleotide is going to be the same. That was the ribose, or the deoxyribose in this case, and the phosphoric acid. So we only put the nitrogenous base. As you could see, these are obviously complementary bases. You have here adenine, so the strand that goes below has to be thymine, adenine, cytosine, guanine, always the same. Complementary basis is essential for you to know. So we open these two strands, and from one of them, just one of them, we are going to produce just one single strand of messenger RNA. So this enzyme is going to be the worker that is going to produce the messenger RNA. How is that going to happen? Exactly the same as replication, for example. We have to do it through complementary bases. So, but taking into account one thing. Remember that when we said complementary bases, we were talking about this. Right? But now, as we are going to produce a messenger RNA, no thymine we have to get. What do we have to get? A uracil, right? So we have to substitute the thymine that we have to put for a uracil. Let's see it. For example, you have here this strand, and we are going to do this RNA thanks to this strand. So, for example, you have a cytosine here, so the following nucleotide has to have guanine, right? Here you have thymine. Okay, so you have to have here the complementary one, that is adenine, right? Here, what does it happen? We have adenine, the complementary basis. Usually is thymine, but as we are producing an RNA, what is going to be? Exactly, a uracil. Again, thymine, adenine again. So the complementary basis, uracil again, right? And we continue until we stop doing the transcription. There are some codes to stop that, okay? And it stops the transcription, and the molecule of messenger RNA is released. No changes have happened in the DNA. It's just that we have produced one molecule of messenger RNA. This messenger RNA is going to mature, and it's going for the next things that are going to happen to it is that it has to mature a little bit, but that is not something that we are going to study this year. And then it will go out of the nucleus, okay? And then when it gets out of the nucleus, the other process happens, which is translation. Okay, I hope that transcription is clear. If you have any question, just ask. Okay, bye-bye.